All right, so today we're diving into something pretty cool, cyclodextrins. But we're not talking about the usual stuff with small molecules, the things they're typically known for. We're going deeper.
Yeah, we are.
You sent over this really interesting video transcript from CarboHyde, and it got me thinking.
Good.
We need to unpack how these cyclodextrins are shaking things up in the world of biologics. Like, what are the big trends? Where's all this heading?
Definitely a lot to unpack.
It's kind of wild to think these little, donut-shaped molecules are at the forefront of cutting-edge biotech.
It is pretty wild.
So for anyone listening who might not be totally up to speed on cyclodextrins,
right?
Can you give us the quick rundown?
Yeah, so cyclodextrins, they're these cyclic molecules. Think of them like rings or, as you said, donuts. They've got this key central cavity.
Donut hole.
Exactly, donut hole. And they're actually made up of glucose units, which come from starch.
So they're basically sugar molecules.
Kind of, yeah. They're naturally derived. But the really neat thing is we can chemically modify them.
Okay.
So scientists can tweak their structure, create a whole bunch of different cyclodextrin derivatives, each with their own little quirks, and we can tailor them for specific uses.
That's pretty handy.
Very handy.
So we've got these molecular donuts made from sugar. Why all the fuss? What makes them so special?
So it all comes down to that central cavity, that donut hole.
Right.
It doesn't like water. It's hydrophobic. And that lets it trap other hydrophobic molecules inside, at least temporarily.
Oh, okay.
And this little hideaway can actually change the guest molecule's properties.
Interesting.
Like traditionally in pharmaceuticals, we've used them mainly to make drugs that hate water dissolve better, to stabilize them, even to get rid of nasty tastes. Mostly, this has been with small molecule drugs, your classic medications.
Yeah, I get it. It's like giving a shy molecule a little safe space to hang out in.
Yeah, something like that.
But now, these cyclodextrins are moving on to bigger and better things. They're interacting with these huge, complex biological molecules.
Biologics.
Biologics, yeah. And that's where things get super exciting.
Definitely exciting.
The CarboHyde video really nicely breaks down cyclodextrin's impact on biologics into 4 main areas. We're going to look at how they work with proteins, their role in vaccines, some other cool uses in biotech processes, and then the really futuristic stuff, their potential in gene delivery.
Sounds like a plan.
So proteins first. When it comes to proteins, it seems like cyclodextrins are doing something quite different compared to their work with those smaller molecules.
Yeah, it's a different ballgame.
I mean, sure, they can still help with solubility a bit, but the real game changer here is how they interact with these massive, intricate structures.
You're talking about your monoclonal antibodies?
Yeah.
Oligopeptides? Even insulin?
It's less about making them dissolve and more about something else entirely. What are cyclodextrins actually doing with these larger protein molecules?
So the big thing is they're preventing protein aggregation. Proteins, they love to clump together. It's in their nature.
Like a big molecular party.
Kind of. But that's a problem. It can make them useless as medicines.
Makes sense.
And it can even set off nasty immune reactions in the body.
Oh, that's no good.
Not good at all. The amazing thing is, cyclodextrins can step in and stabilize these proteins. They're like tiny shields.
Preventing the party.
Exactly. They stop the proteins from getting too close and forming those clumps, those aggregates.
So it's all about keeping those proteins from misbehaving.
You got it.
Are there any perks beyond just preventing these protein pileups?
Oh, absolutely. The transcript mentions a bunch of added bonuses. For example, by stopping aggregation, cyclodextrins can actually make protein solutions less viscous, less thick.
Okay, I see.
This is a big deal for injectables. It makes them way easier to administer.
Makes sense. Nobody wants a thick, gloopy injection.
Nope. They can also stop proteins from sticking to the walls of containers or, the devices we use to deliver them.
Right, so the patient gets the full dose.
Exactly. And sometimes they might even boost bioavailability, meaning the protein gets absorbed better and reaches its target more effectively.
Wow, that's impressive. So these cyclodextrins are like the ultimate protein chaperones.
They are pretty good at their job.
It sounds like keeping protein stable is a major headache in the world of biologics. What are the main challenges, and how do cyclodextrins help?
So proteins are fragile. They're susceptible to all sorts of things.
Like what?
We've talked about aggregation, but there's also denaturation. That's when they lose their specific 3D shape, and that shape is crucial for them to work properly.
Right.
Then you've got enzymatic degradation, where enzymes break them down.
Yeah.
And even oxidation, which is like a chemical attack.
Oh.
All these things can be triggered by changes in temperature, shaking them up too much, even just time.
So storage is an issue, too.
Oh, yeah. Now, the transcript makes it clear that cyclodextrins aren't a cure-all. They won't solve every protein problem in every situation, but there's a growing pile of evidence that they can handle a lot of these stability challenges.
That's good to hear. Let's get down to specifics. Are there certain cyclodextrins that are being used or showing real promise in these protein applications?
The transcript focuses on 2 that are approved for parenteral use. That means that they're safe to inject.
Okay.
These are hydroxypropyl beta-cyclodextrin, or HPBCD for short,
HBCD.
and sulfobutylleather beta-cyclodextrin, SBCD.
SBCD. So those are the big players when you're formulating injectables.
Yep, they've got the best safety records.
It also seems like there are companies and research groups really dedicating themselves to this area.
For sure. The transcript mentions Kleptos Biopharma, and they actually offer a special grade of HPBCD specifically for biologics.
So they're all in on this.
They are. And because of that, they're a major source of the data we have on HPBCD and how it performs with proteins. The transcript also highlights studies from Rocket Pharmaceuticals showing they're actively exploring HPBCD as well.
This next part caught my attention, the synergy between cyclodextrins and those classic stabilizers.
Yeah.
Tell me more about this dynamic duo.
This is where things get really interesting. So we've got cyclodextrins and polysorbates. Polysorbates are like the old, reliable stabilizers in protein formulations.
It's veterans.
Haha, exactly. And at first people thought, well, maybe cyclodextrins could replace them entirely, which makes sense because they're safer, they don't get broken down in the body, they don't create those nasty reactive oxygen species.
They're the cleaner option.
Yeah, definitely. And they can replace polysorbates in many cases. But the researchers at Rocket Pharmaceuticals found something amazing.
What's that?
They found that when you combine them, a cyclodextrin and a polysorbate, the reduction in protein aggregation is way more dramatic than you'd expect from just adding their individual effects.
Wow. So 1 + 1 = 3 in this case.
Exactly. It's a synergistic effect, and it probably happens because they work in different ways at a molecular level. It's like a double-pronged attack on protein instability.
2 security systems are better than 1.
You got it.
Now, the transcript gets into the nitty-gritty of how cyclodextrins interact with proteins. They use insulin as an example. And it's not that simple 1-to-1 encapsulation we see with smaller molecules, right?
No, not that simple. Proteins are giants compared to cyclodextrins. Their surfaces are way more complex.
Right.
So you don't usually get one protein snugly fitting inside one cyclodextrin cavity. It's more like they're dancing around each other.
A molecular waltz.
Haha, I like that. Imagine multiple cyclodextrins interacting with different amino acids on the protein's surface.
Okay.
Some parts of the protein might peek into the hydrophobic cavity, but you also get these weaker bonds forming, like hydrogen bonds with HPBCD or electrostatic attractions with SBCD.
So it's all about finding the right fit.
Exactly. How well a particular cyclodextrin stabilizes a protein depends on how well they can shake hands, so to speak.
If they don't click, it's not going to work.
Pretty much.
The transcript mentions a ton of research on cyclodextrins and protein formulation. It seems like they're becoming the go-to excipient, the helper ingredient.
They are definitely gaining popularity. There are tons of studies on monoclonal antibodies and other therapeutic proteins, all pointing to cyclodextrins as a way to improve their stability and make them easier to formulate.
It's good news for protein-based drugs.
Definitely.
This next concept is really cool. Cyclodextrins forming a protective layer around a protein, like a self-assembling shield. How does that work?
This is where things get futuristic. Instead of individual cyclodextrins or even the usual polymers, you have these chains of cyclodextrins spontaneously wrapping themselves around the protein.
Wow.
It's like they're building a custom-fit suit of armor.
Impressive.
And the forces holding it all together, that could be a combination of those little protein bits fitting into the cavities and weaker interactions on the surface.
So both types of interaction working together.
Exactly. And this approach would be super specific to each protein's unique shape, which is pretty cool.
It is. Now, what about some of those practical problems in protein formulation? We've talked about viscosity, and there's also the issue of proteins sticking to containers. How do cyclodextrins help with those?
Right, so viscosity. Basically, the main reason protein solutions get thick is because they clump up.
The aggregation we talked about.
Exactly. Cyclodextrins stop that clumping, so the solution stays nice and flowy.
Okay.
Interestingly, the cyclodextrins themselves don't really add much to the viscosity, at least not at the concentrations we normally use.
So they're not making the problem worse.
Nope. It's only at crazy high concentrations that their own viscosity becomes noticeable.
Interesting. And what about the sticking to containers?
It's probably a multi-pronged attack. Cyclodextrins are abundant, so they can compete with the protein for those sticky spots on the container. Plus, they don't stick to each other, so they don't form those protein-attracting layers on the wall. And they can even interact with the protein itself, maybe covering up the parts that like to stick.
Like a molecular camouflage?
Yeah, something like that.
Despite all this awesome research, it seems like there aren't a ton of cyclodextrin-based protein drugs on the market yet. The transcript mentions a couple of examples.
Yeah, you're right. The research looks fantastic, but there are only a few commercial products so far.
What are they?
The transcript talks about Kyprolis. It uses HPBCD.
HPBCD.
But it's a pretty small peptide, just 4 amino acids long. So it acts more like a small molecule, and the cyclodextrin mainly helps with solubility there.
Interesting.
A better example is Baqimi. It's a glucagon nasal powder, and it uses beta-cyclodextrin.
Oh.
Now, because it's delivered nasally, not injected, there's more flexibility in terms of which cyclodextrins you can use.
I see.
Eli Lilly chose beta-cyclodextrin, which shows it's a good fit for this kind of application.
And what about the future? Are there more cyclodextrin-containing protein drugs in the pipeline?
Oh yeah, the good news is there are a bunch of products in preclinical development.
So things are moving.
Definitely. As those promising candidates go through the hoops of drug development, I think we'll see a lot more cyclodextrins used for protein stabilization in the next few years.
That's exciting.
It is.
The transcript also briefly mentions antibody cyclodextrin conjugates. What's the deal with those?
So this is a newer approach where you directly attach cyclodextrins to monoclonal antibodies.
Interesting.
The goal is to make the antibody better, maybe make it last longer in the bloodstream, help it target the right cells, or even change how it interacts with its target.
So it's like giving the antibody a power-up.
Exactly. There's definitely research going on, but it's still early days.
Okay, let's move on to vaccines. It seems like cyclodextrins have been around in this field for a while. Longer than in protein therapies.
They've got a longer history, which might surprise some people. Pfizer was a real pioneer in this area, especially with vaccines for animals.
Vaccines for animals. Tell me more about that.
So the transcript highlights 2 products for veterinary use. The first one is Suvaxyn. It was actually the very first product to use a cyclodextrin, not just as a formulation aid, but as a vaccine adjuvant.
A vaccine adjuvant? What's that?
It's something you add to a vaccine to boost the immune response, make it work better.
Ah.
And the cyclodextrin they used in Suvaxyn is called sulfonyl ether lipocyclodextrin.
That's a mouthful.
It is. Suvaxyn was developed back in the 90s. And they've been using this specific cyclodextrin as the adjuvant ever since, but here's the weird part. No other product uses it for that purpose.
Really? So it's a one-hit wonder?
Pretty much. The second example is a vaccine for Newcastle disease in birds. It uses hydroxypropyl beta-cyclodextrin, or HPBCD, the same one used for protein stabilization.
Oh, interesting.
And they use it as an adjuvant, too.
So it's a multitasker.
It is. This was a big deal because it showed that a common excipient used in injectables could also boost the immune response.
That's cool. So what about human vaccines? Are cyclodextrins being used there, too?
Yep. They are. The transcript mentions a nasal flu vaccine that's been tested in clinical trials. It uses HPBCD as an adjuvant.
Nasal flu vaccine.
Okay. And the results were interesting. They saw a small but noticeable increase in the T-cell response, suggesting it was boosting immunity right there in the nose, where the vaccine is given.
Makes sense.
This could potentially reduce allergic reactions and give longer-lasting protection.
That'd be great.
It would. And it makes you wonder if the way cyclodextrins work as adjuvants is similar to how they work with small molecules.
Good question. It sounds like researchers are working on designing even better cyclodextrin-based adjuvants.
Oh, yeah, definitely. CarboHyde, the ones who made that video, they're right at the forefront of this research.
What are they doing?
They recently did a big study where they screened a huge library of cyclodextrins to see how they affect immune cells, specifically monocytes and lymphocytes.
Important cells for immunity.
Exactly. And they measured all sorts of markers. The results were pretty amazing. All the cyclodextrins were safe, but they triggered different levels of immune response.
Different levels.
Yeah, some were way more potent than others. Some even outperformed the positive control, which was E. coli lipopolysaccharide. That's a really strong immune stimulant.
Wow, that's impressive.
It is, and this research is crucial for understanding how the structure of a cyclodextrin influences its adjuvant activity. The goal is to create a new generation of super effective adjuvants.
Adjuvants 2.0.
Exactly.
So beyond adjuvants, are there other ways cyclodextrins are being used in vaccines?
Absolutely. Since they're so good at stabilizing proteins, it's no surprise they can stabilize the antigens in vaccines.
Antigens?
Yeah, those are the protein bits that trigger the immune response.
Ah, all right.
The transcript gives an example of a Sanofi Pasteur patent where they used cyclodextrin in a polio vaccine. It stopped the virus from degrading because of the preservatives.
Clever.
Another cool application is in cryopreservation. That's basically freezing stuff without ruining it.
I've heard of that.
The Johnson & Johnson COVID-19 vaccine actually contained HPBCD.
Oh, really?
Yeah. And the transcript suggests it might have acted as a cryoprotectant during storage.
So it kept the vaccine happy in the freezer?
Exactly. This cryoprotective effect is used a lot in vet medicine and research.
Like for storing cells or?
Yeah, things like cell cultures, sperm, you name it. They often use methylated cyclodextrins for that. These modified cyclodextrins are pretty cool because they can stabilize things and protect them from freezing damage.
Double duty.
Yep. And cyclodextrins can also help maintain the chemical integrity of certain vaccine components over time.
So they're like the all-around helpers in vaccine formulation.
They are pretty versatile.
Okay, let's zoom out a bit. Beyond these direct applications in therapies, how are cyclodextrins being used in the wider biotech industry, like in manufacturing processes?
This is a really cool area. People don't always think about it, but cyclodextrins are being used behind the scenes to make all sorts of biotech products.
Like using bacteria or enzymes to produce stuff.
Exactly. And what's exciting is that a lot of these applications aren't just lab experiments. They're already being used commercially.
So how are cyclodextrins helping to improve these biotech processes, like making them more efficient or boosting production?
There are two main ways they contribute. First, they can help dissolve starting materials at much higher concentrations.
Like the raw ingredients?
Yep, the chemicals, nutrients, whatever you need.
Okay.
And by making them more concentrated, they're more readily available for enzymatic reactions or for microbes to use. This can really speed things up.
Makes sense.
Second, sometimes in these bioproduction processes, the product itself can actually hinder its own production. It's like a self-sabotage situation.
That's frustrating.
It is. But cyclodextrins can come to the rescue. They can grab those inhibitory products and trap them in their cavities, effectively removing them from the equation. This allows production to keep chugging along, and you end up with a higher yield.
It's like cleaning up the clutter so the work can continue.
Haha, exactly. The transcript mentions an example where complexing cholesterol with cyclodextrin improved production.
Interesting. Do you have a more concrete example, like a specific process where this is being used?
Absolutely. Take the production of L-asparaginase, for example. It's an enzyme used to treat certain cancers.
Okay.
Research has shown that adding cyclodextrins to the mix, especially beta-cyclodextrin, can significantly increase the yield of this enzyme.
So more bang for your buck.
Exactly. And the cool thing is that this often happens without affecting the growth of the microbes that are producing the enzyme.
So it's a win-win.
It is. And sometimes cyclodextrins can even act as a food source for microbes, making certain nutrients more easily accessible.
That's pretty neat.
It is.
The transcript specifically mentions the production of pertussis toxin, which is used in the whooping cough vaccine. Can you tell us about that?
Yeah, this is a great example. So during the production of pertussis toxin, you get these fatty acid byproducts that actually start to slow down the whole process.
They gum up the works.
Haha, exactly. And this is where a special type of cyclodextrin comes in called DPA.
DPA. Another mouthful.
Haha. It stands for heptakis 2006 dimethyl beta-cyclodextrin.
Catchy.
Right. So DMA can selectively bind to those pesky fatty acids and keep them out of the way.
It's like the bouncer at the molecular club.
Haha, love that analogy, and it works incredibly well. The transcript says it boosted toxoid production over 100-fold at a concentration of just 1%.
Wow, that's remarkable.
It is. It shows how choosing the right cyclodextrin can make a huge difference.
So what's so special about this DMA? It's another methylated beta-cyclodextrin, right?
Yeah, but it's not your average methylated beta-cyclodextrin. You might have heard of the randomly methylated kind, which is used for all sorts of things.
Right.
But DMA has a very specific pattern of methylation. It's only methylated at certain positions on the glucose units, not just randomly thrown in there.
So it's a precision tool.
Exactly. And this makes it much harder to produce, which is why it's more expensive.
Makes sense.
The transcript even mentions that there's often confusion about its name, which can make it tricky to find the right product.
So it's a bit of a hidden gem.
You could say that.
All right, let's move on to our final topic, gene delivery. It sounds like this is the newest frontier for cyclodextrins, with tons of potential but still in the early stages.
Definitely the newest frontier. There's a lot of excitement, but it's all preclinical research so far. Nothing's reached human trials yet.
So what are the big challenges in gene delivery, and how might cyclodextrins help?
It boils down to two things. First, you have to protect the genetic material, whether it's DNA or RNA, from getting chopped up by enzymes.
Right, it's fragile stuff.
It is. And second, you have to make sure it gets into the right cells. And not just into the cell, but into the specific part of the cell where it can do its job.
So it's like a targeted delivery mission.
Exactly. There are different systems being developed to do this, some using viruses and some not.
Viral and non-viral.
Yep. Non-viral methods are getting a lot of attention because they're potentially safer.
Makes sense.
At first, people thought about using cyclodextrins as parts of existing delivery systems, like liposomes or nanoparticles.
Like adding them to the mix.
Right. The idea was that cyclodextrins might make those systems less likely to trigger an immune response, basically making them safer.
Okay.
But now, researchers are exploring whether cyclodextrins can actually deliver genes themselves.
So taking center stage.
Exactly. And there are a few different approaches. One is to create cationic polymers of cyclodextrins. These are positively charged, so they can interact with DNA, which is negatively charged.
Opposites attract.
Yep. They form complexes that cells can then take up. The transcript mentions some work with Dayton University on this.
Interesting.
Another strategy uses polyrotaxanes. These are weird structures where cyclodextrins are threaded onto a polymer chain, kind of like beads on a string.
I'm picturing it.
And these polyrotaxanes can also interact with DNA.
Cool.
Then you've got amphiphilic cyclodextrins. These have both hydrophobic and hydrophilic parts, so they can self-assemble into nanoparticles in water.
Nanoparticles are all the rage these days.
They are. And these nanoparticles can encapsulate the genes. What's interesting is that the cyclodextrin cavity itself isn't necessarily involved in this case.
So it's the nanoparticle doing the work.
Yep. And finally, there's some cool research from Sorbonne University on self-assembling cyclodextrins. Here, the individual cyclodextrins spontaneously organize themselves around DNA or RNA, protecting it and helping it get into cells.
That's amazing.
It is.
Let's dig a bit deeper into those cyclodextrin polymers and amphiphilic cyclodextrins. What makes them so good for gene delivery?
Cyclodextrin polymers are incredibly versatile. You can design them with different charges, make them biodegradable or not, and they're generally good at forming complexes with DNA.
It's like a build-your-own gene delivery system.
Kind of. Right now, they're mostly used for things like wastewater treatment.
Oh, wow.
But they have huge potential in pharma. You can even add specific molecules to them that target certain cells, making them even better at delivering genes.
That's clever. And what about the amphiphilic cyclodextrins?
These are great for making nanoparticles. They can condense DNA into their core, keeping it safe and sound.
Tiny protective bubbles.
Exactly. The GenCat consortium has been working on these. They've created a huge library of amphiphilic cyclodextrins and tested how well they deliver antisense oligonucleotides.
Antisense oligonucleotides.
Those are short DNA or RNA sequences that can interfere with gene expression.
Oh, okay.
And they've found that the structure of the cyclodextrin really influences how well it works.
So subtle changes make a big difference.
They do. One of the most exciting things is that these cyclodextrin nanoparticles seem to be safer than lipid nanoparticles, which are another popular non-viral delivery system.
Safer in what way?
They don't trigger as much of an immune response, which is a big plus.
It's important.
And they can deliver genes just as effectively. They can even package different sizes of oligonucleotides.
So they're versatile and safe.
Exactly.
The transcript even hints that they could be used to deliver other things, not just genes.
Yep. Maybe even small molecules or other biologics.
That's really cool.
It is.
Well, this has been a fascinating journey into the world of cyclodextrins and biologics. It's amazing how these little donut-shaped molecules are making such a big impact.
They are pretty amazing.
Any final thoughts before we wrap up?
So I think the main takeaway is that cyclodextrins are way more versatile than people might think. They're not just for small molecules anymore. They're already being used in vaccines, in biotech manufacturing, and protein stabilization is definitely on the horizon. Gene delivery is further out, but it holds huge promise.
And it sounds like we need new and improved cyclodextrins for those gene delivery applications.
Yep, lots of exciting chemistry to be done.
It's incredible how these simple molecules can have such a profound impact on such complex areas of biotech and medicine. It makes you wonder, what else can they do? What other hidden talents are waiting to be discovered?
Who knows what the future holds?
Right? Maybe they'll be delivering CRISPR therapy someday.
Maybe. Or nanobots.
Now you're getting carried away.
Maybe. But seriously, it seems like we've only just scratched the surface of their potential. There's so much more to explore.
I'm definitely intrigued. To everyone listening, we encourage you to check out the research we've discussed today. It's a fascinating field, and there's always something new to learn. Thanks for joining us on this deep dive.
Thanks for having me.